We've talked about amino acids, and now we're going to talk about peptides. Specifically, we're going to define polypeptide and identify the amide bond. Peptides are what form when amino acids bond together. And one thing we have to worry about are the terminal ends. We have the N terminus of an amino acid, and we have the C terminus of an amino acid. So obviously the N terminus is the amino acid end, and the C terminus is the carboxylic acid end. Notice that the side chain is never considered the N or the C terminus, regardless of the composition of the side chain. The termini have to be in the backbone of the amino acid. And this becomes important because as we start to bond amino acids together, they have to be bonded in a specific way. Let's look at the simplest of peptides, which is a dipeptide, which is 2 amino acids. So peptide is just another term used for amino acid. When we look at this dipeptide, what we're going to look for is try to identify the backbone and the different amino acids that make it up. In this case, we have our amine group, our CH, C double bond O. We have our amine group, our CH, our C double bond O. In this case, there is no oxygen in this one with our carboxyl group because it went away when we had the condensation reaction to form it. As a result, all of this is considered the backbone of the dipeptide, and then we have 2 side chains. If we look at where the bond formed here, which is also where it will break in hydrolysis because those are opposite reactions, we will see that it's right between the carbonyl and the nitrogen. So we have NCC, NCC. That's the pattern we're looking for in our peptides. Another flashback to functional groups. Which one is an amide functional group? Hopefully you picked B, okay, because an amide group has a nitrogen bonded to a carbon that has bonded to a double bonded to an oxygen. The reason an amide group is important is because the amide bond is what forms between 2 amino acids. And this is also known as a peptide bond. So we see our nitrogen bonded to a carbon with a double bonded oxygen. And that's an amide functional group, so we call it amide bond. Let's compare these 2 molecules and see if you think they are isomers or the same molecule. When we look at these molecules, the key thing we have to look at is the backbone, because that's where we're going to start our comparisons. So we see our N terminus is on the left side, the N terminus is on the left side. And that means we're going to compare both of these in the same direction. If one of these had been written differently, we would need to start with the N terminus on both of them. So as I go through, I see my N terminus, I see CH, C double bond O, NH, CH, C double bond O, OH. I see the same pattern on the one on the right. The first amino acid in the left molecule is CH3, the first side chain. The second side chain is CH2OH. In my other molecule, it's CH2OH and CH3. These are considered isomers because they are not bonded in the same order. So as long as I start both comparisons from the N terminus and then see what order the side chains are in. If they are in a different order but the same side chains, then these are isomers. Not surprisingly, a tripeptide has 3 amino acids in it. If we look, we can identify where those are. So here's the first one because I have my NCC. Then I have NCC, so there's my second one breaks off, and then NCC with the OH at the end. So these are the 3 amino acids that make up the tripeptide. Polypeptides form in our body, and these are typically known as proteins or enzymes. Poly means many, so many amino acids connected together. From our DNA, information from genes is transcribed into the RNA, which then leaves the nucleus of the cell, and that information is translated to form the proteins and enzymes. And this occurs in the ribosome. So information in our DNA tells our body what needs to be made so that we have the proteins and enzymes we need to function. We won't get into more discussion about DNA here, but you will likely discuss it in biology class if you haven't already. So polypeptides are just sequences of amino acids, and they're a big long chain, so every little sphere here represents a different amino acid. And if we zoomed in on those, we would see the same backbone on every single one, and we have the N-terminus on one end, we have the C-terminus on the other end, and it's always the C-termini and the N-termini that are bonding together to form this polypeptide. How many amino acids formed this polypeptide? Okay. To identify this, we want to look for our backbone, the NCC pattern. So we have NCC. Okay. There's one. NCC. There's another one. NCC. And another one. NCC. So we have 1, 2, 3, 4 amino acids that make up this polypeptide.